T helper cells lie at heart of all immune responses. They occupy a central place, extending their head to various wings of immune response, innate, humoral, cell-mediated immunity. सबको ये help करते हैं. How that happens, we are going to discuss now, and that will more explicitly cover one more topic also, how various wings of immune response they interact with one another, at center of which lie the T helper cells. They are lymphocytes, T lymphocytes. Why are they called T lymphocytes? Like every type of hemopoietic tissue, their origin is in the bone marrow. The progenitor cells, they originate in the bone marrow. Bone marrow say they go to the thymus, and they undergo what we call thymic education. They undergo maturation within the thymus, and when they emerge from the thymus, they are mature. Mature means they are immunocompetent. What does it mean, immunocompetence? It means that they have acquired certain receptors which are called T cell receptors, abbreviated as TCR. So, T helper cells, they become immunocompetent. Immunocompetent means they are competent enough to elicit, to participate in the immune response now. So, we are going to see how this happens. We start with the same thing, a pathogen entering the body. This is a pathogen, it has entered, this has crossed the skin barrier, breached the skin barrier and entered the body. First of all, it will be confronted by macrophage, for example, a phagocytotic cell. Macrophage comes into picture, abbreviated like this. Now macrophage is going to internalize this, और internalize करने के बाद macrophage is going to degrade it with the help of its lysosomal enzymes. Macrophage will degrade the invading pathogen into various very small antigenic fragments. अब ये जो antigenic fragment है, these will be presented on surface of the cell with the help of antigenic fragment. I am showing in red. These are certain molecules which are called MHC molecules. MHC means major histocompatibility complex. So in major histocompatibility complex, these are certain heterodimers which are presented on the cell surface. They have got a groove, and into this groove one of the antigenic fragment is presented. So the primary role of MHC, major histocompatibility complex, is to transport and present the antigen on the cell surface. I say this because earlier their role was primarily thought to be in transplantations, etc. But now it is well known that their primary role is to present the antigenic fragment, and accordingly the macrophage is also called antigen presenting cell. There are some other types also. Please find out what are they. So up till now we have seen pathogen, internalized, degraded, antigenic fragment now on the surface of the cell. अब क्या होगा? We are going to see now. Now the T helper cell will come into picture. T helper cell is going to now recognize this antigenic fragment in association with MHC. I repeat, T helper cell का जो TCR है, T cell receptor, it can recognize antigenic fragment only when it is in association with MHC. And MHC also various types are there, MHC2. So this provides a stimulatory signal for the T helper cell. Now the T helper cell, it will start proliferating. Stimulus ho gaya, so it will start proliferating. So this is the first signal which we have discussed till now. I am abbreviating it as SS, it means stimulatory signal. Alright, now comes into picture a second signal which is called cytokine signal. Cytokines are soluble mediator of all immune responses. They are very small molecular weight and they have got a very short half-life, and they control various cells of various rings of immune response. As we are going to see, they have got autocrine function, paracrine functions, etc. etc. So cytokine signaling, now we are coming to, I will just know, cytokine signaling, the second one. Now there is activation not only of the T cells but also of the macrophages because of this interaction of MHC, antigenic fragment, and the TCR. As a result, from macrophages is secreted a cytokine called interleukin 1. Interleukin 1, it's a cytokine. So by its paracrine action on the T helper cell, it will activate the T helper cell, of course, and the activated T helper cell will start secreting another cytokine called interleukin 2. Interleukin 2, it has got an autocrine function. It acts on the same cell which secreted it, that is the T helper cell. In fact, an interesting point, they occupy a receptor on the T helper cell, and upregulation of this receptor is again caused by interleukin 2. So with these effects, the T helper cells is going to undergo two changes. First, the T helper cell, the activated T helper cell, is going to proliferate like anything. This process of proliferation is also called clonal expansion. So proliferation, number one, and the second is polarization. What does it mean? The proliferated cells, they differentiate into various phenotypes like TH1, TH2, TH17, TH regulatory, various phenotypes they differentiate into. This is called polarization. So first activation with the stimulatory signal IL-1, then IL-2 comes into picture with the result. The ultimate is clonal expansion. Number two, polarization. Now the TH1, they continue to secrete interleukin 2, whereas TH2, they shift on to production of interleukin 4 and interleukin 5. So अभी तक हमने देखा TH1, they start secreting now interleukin 2, and then TH2, they shift on to production of interleukin 4 and 5. They secrete not only 2, they also secrete interleukin 12. So both these, interleukin 2 and interleukin 10, they stimulate the cell-mediated immunity. And interleukin 4 and 5, they stimulate the humoral immunity. As you all know, humoral immunity, B cell say antibody, B cell differentiates into plasma cell and then antibodies are secreted. Interleukin 4 and 5 stimulate this process. And cell-mediated immunity, as you know very well, it takes care of the intracellular pathogen. So it is mediated by the T cytotoxic cell. Under influence of interleukin 2 and 12, there is burst in activity of the T cytotoxic cells. burst in proliferation, their activity, as a result of which they are able to take care of the intracellular pathogen. One more point here. So here we have seen how they are affecting the cell-mediated and the humoral immunity. Th1, they also secrete what we call another cytokine, interferon gamma. And this enhances activity of the macrophages, the microbicidal activity of the macrophages. Please recall the same figure, मैक्रोफेज टी हेल्पर सेल. अब ये जो मैक्रोफेज था उसकी एक्टिविटी भी बढ़ गई. सो वी हैव टॉक्ड अबाउट टू सिग्नल्स टिल नाउ. प्लीज नोट, द फर्स्ट वन वाज स्टिमुलेटरी सिग्नल, द सेकंड वन वाज साइटोकाइन सिग्नल. तो स्टिमुलेटरी है तो एक को स्टिमुलेटरी सिग्नल भी होना चाहिए. आई एम राइटिंग इट एज OS. तो ये क्या होता है? दिस इज विद द हेल्प ऑफ सर्टेन CD molecule, cluster of differentiation molecules. अब जैसे हमने देखा था मैक्रोफेज एंड टी हेल्पर सेल, तो इसके सरफेस के ऊपर CD, a glycoprotein, and a counter ligand of this is present on the surface of the T helper cell also. I think इनके नाम है, this is B7 and this is called CD28. Alright, so this provides the additional signal, the co-stimulatory signal. Just to know a question for you, if antibodies are raised against B7, what is going to happen? The immune response is going to flounder, it is going to be attenuated, becomes very weak. Why? Think about it. So the three signals we have talked about, और हमने देखा T helper cells, they lie at core of all activities, mainly through cytokines, so many cytokines affecting so many different processes. Thank you so much.